Okay, good. I'm Melissa Geller, a GYN oncologist here at the University of Minnesota, and it's my pleasure to introduce today to you Dr. Kristina Thiel. She's an assistant professor of obstetrics and gynecology at the University of Iowa and is a member of the Holden Comprehensive Cancer Center there. She's a molecular cancer biologist whose work is really focused on translational research and focusing particularly on P53 biology, as well as endometrial and ovarian cancer therapeutics, as well as patient-derived organoid models. And I asked her if she knit this, but she didn't. And I thought that's very near and dear to my heart, the uterus and the ovaries. So Dr. Thiel completed her PhD in biochemistry at Vanderbilt University and postdoctoral training at the University of Iowa, where she is currently faculty there. She is a co-founder, I found very interesting, of a Iowa spinoff company that looks at AI-driven approaches to personalized cancer treatment. She's won multiple innovative awards for that, including and was elected to the Iowa chapter of the National Academy of Inventors. Her research currently is supported by a K-22 Career Development Award, as well as she's running a Project One on a NCI-funded PO1, looking at advancing hormone therapy for endometrial cancer. She holds multiple DoD grants as well as institutional grants. So I'd like to welcome her here today, and her talk today is going to highlight some of the endometrial hormonal therapy that we have options for currently in GYN cancer. Thanks.
Okay, well, thank you so much, Dr. Geller, for that really kind introduction. It's we talked at breakfast this morning about imposter syndrome, and I will say sometimes it's hard to hear a really glowing introduction of yourself and you think, is that really me? And then you have to get up and give a talk and you think, is my data going to live up to that? So hopefully what I'm going to show you today is interesting and exciting, and we'll just get your wheels turning about how we can advance treatments for endometrial cancer. So I do have an active research program in ovarian cancer, but just for the sake of a kind of a complete talk today, I'll only be talking about our published and mostly ongoing unpublished work in endometrial cancer. So, you know, please don't put these ideas into your grants and compete with me. And if you happen to be a reviewer of our paper, please be kind. So before I get started, I wanted to acknowledge that I was brought here through the Emerging Leaders Exchange through cancer centers in some of the Big Ten institutes. At the University of Iowa, we have a really robust Emerging Leaders Council through the Holden Comprehensive Cancer Center. I was brought in, I was in the inaugural cohort of the ELC, we call it. So I'm now a emerged leader. I've since graduated. This is the third cohort currently. And just really what I want to highlight is it spans 3 different colleges, liberal arts and sciences, medicine, which you can imagine most of the faculty draw from, and nursing. And we're hoping to encompass all of the colleges across the campus as we move forward. If you look at some of the titles here, you see that we have emerging leaders that are tenure track faculty, clinical faculty, and research faculty. So we're not exclusive to tenure track faculty. This provides leadership opportunities for people at different, that have different career goals and leadership goals. And then the leaders are drawing from the 3 different programs within the Cancer Center at University of Iowa: Cancer Genes and Pathways, that's CGP, Experimental and Redox Therapeutics. So as an emerged leader, I'm a associate co-lead for the ERT program, and then cancer epidemiology and population science. So, should I take a quick break if people want to get donuts, or feel free to get up and get donuts? I have kids, you're not going to offend me at all. We can all multitask, right? So really, I just wanted to highlight the incredible opportunities that I've been given through the Emerging Leaders Council to get to understand the inner workings of running a cancer center, and really getting on the radar of the leaders and my own career aspirations being fostered through that. But I think most importantly is I got to know other junior faculty that were coming in my same career stage. And so we formed a really fantastic group where we meet regularly. even beyond Emerging Leaders Council, to share challenges in running a lab and grant funding and celebrate successes. And I hope that, if there's inspiration I can share, that this has just been a great opportunity. Okay, without further ado, let me get into what I'm going to be talking about today. So first I'll go over a little bit of background on uterine cancer, endometrial cancer. So the endometrium is the lining of the uterus, and then how that's been treated with hormone therapy over the years, and then discuss some of the barriers to continuing to advance hormone therapy as a treatment for endometrial cancer, and then some of the approaches that we're doing to identify better treatment options. Okay, so it's not a cancer slide talk without this slide. The 2025 ACS facts and figures, 26 will drop in a couple of months, but every year for the last however many decades of my career, it comes out and it's all the same. We know that uterine cancer is the most common gynecologic malignancy in women, and ovarian cancer is the most deadly. Until 2024. And now uterine cancer has surpassed ovarian cancer as the most deadly gynecologic malignancy. And this is really a call to action that we need better research into understanding what's driving this increase in endometrial cancer deaths. And so if you look at the 5-year survival rates for all cancer types in the US, for most of these, let's see if my pointer, you guys see the pointer there? Yeah. The arrow is going to the right, which means we're improving survival. Prostate's getting better, thyroid, melanoma, breast, and oh, wait a second, uterus. That arrow is going in the wrong direction. So our patients are dying more now than they were in the 70s. And the only other cancer type for which there is a decreased survival is cervical cancer. So why is survival for endometrial cancer going in the wrong direction? So there are lots of hypotheses. I could give a whole talk on this, but my personal professional opinion is that we have a lack of really fantastic models that replicate the heterogeneity of disease in patients. So in breast cancer, for example, there are syngeneic models. There are genetic models. There are lots of patient-derived xenograft models. But for endometrial cancer, there are 2 cell lines that represent early stage grade endometrial cancer. So what our lab does is we really focus on using patient samples to better replicate the heterogeneity within a patient as well as across patients. So you all know about cell lines. They're the workhorse of cancer research. They are easy and inexpensive to maintain, although it's pretty challenging to get a tumor from a patient to grow in plastic. They evolve. And by that reason, they may not represent the clinical spectrum. And I will say, I'm going through this in detail because for the students in my lab, I really feel like we need to understand the strengths and weaknesses of all the models we study and how we can learn from the different models. So it's not a one size fits all and we only use one model. But one great thing about cell lines, as we all know, is you can do high throughput assays in them, which is really in contrast to xenograft models, where you take a tumor and you implant it typically in an immunocompromised mouse, although now we have more humanized mouse models available. So the pro of the xenograft is that it captures that interpatient and intra-patient heterogeneity. And you can understand the tumor microenvironment niche and how you get that complex interplay to allow tumor development, progression, response to therapy. But the success rate is really low. So for most endometrial tumors, it's about 30%. So it's very hard to make them. And they're, of course, expensive to maintain. So kind of the intermediate between a cell line and a xenograft is an organoid model. Has everyone here heard of an organoid model? You're going to hear a lot more about them. Okay. And so they're fairly easy to create as compared to a PDX model. Our success rate is probably about 75%. And they capture the heterogeneity, which is great. They are not nearly as cheap as cell lines. So because they have their special slurry of culture reagents, and you can't just take FBS and put it in DMM or RPMI. It's quite expensive to make it, but they're amenable to high throughput assays. I will say in our hands, organoid models are a finite resource. They won't grow indefinitely like one of my colleagues in the lab next door to mine. She studies colorectal cancer. And most of her models will grow indefinitely, whereas we've seen a genetic drift over time with the model. So we try to get as much data as we can from the first few passages. So to create organoid models, it really takes a concerted effort from multiple different shareholders, including gynecologic oncologists that are helping us as researchers understand what are the cancer types, the histologies, the molecular subtypes that are most in need of better treatments and understanding of the disease pathophysiology. And speaking of pathology, pathology is really integral in providing tumor specimens to us, but that all is really driven by tissue procurement. So what happens is we have a robust tissue procurement core that each week sends us a list of cases that are potentially eligible for tumor collection. And so they know which specific histologies we're interested in. And we tell them yes, no, do we want it fresh? Do we want it cryopreserved, which I'll get to in a second. And then they coordinate with pathology to get the samples from the gross room, from the gross anatomy room, and then transfer it to our laboratory. Our IRB at the University of Iowa is a universal IRB that is applicable to all cancer patients, and we can collect any discarded tumor tissue or any discarded tissue from the patient and it's available for research if the patient consents. If they're having blood draws, we can also get an extra tube. And the great thing about our IRB protocol is it allows for something called post-collection consent. And what that means is that we can collect the tumor tissue and it's stored in the biobank, and we have 60 days, 60 or 90, but I think it's 60, to consent the patient. And this has really been a game changer for our lab because consenting is a challenging process. You have to have staffing available either at the clinic visit or at surgery. So we found that we have pretty good success rate in consenting patients in like day 2 after their surgery while they're still in the hospital and recovery. And they say, yeah, sure, you don't need it for diagnosis. Yeah, you can have the tissue. And then my lab enjoys this too because they can wait for a definitive diagnosis and they're not getting tumor tissue at 4:00 in the afternoon and they have to stay until 7 or 8 at night. So they have, the students like the work-life balance. So we, most of our tissue would get cryopreserved, and we have pretty good success rate creating organoid models from the cryopreserved tissue, although we definitely take a hit in compared to fresh tissue. And then, of course, the laboratory staff that is necessary to create these models, and then all of the great students and trainees I have in my lab that are studying them. Any questions about this process? Because I think I've talked to a lot of colleagues at other universities, and what Iowa has set up is really allows us to collect a lot of models. And I would say we probably get 5 endometrial tumors and 1 ovarian tumor per week. So we're getting a lot of specimens if anyone wants to collaborate. So how do we actually make organoid models? Well, we first start with the tumor tissue, and we slice it and dice it in a little petri dish. And then we digest it with collagenase, disbase, and DNase, and then we use a red blood cell lysis. So it's important to note this because we're not selecting out exclusively tumor epithelia. There are other cell types here, but the culture media, when you plate it in a specialized matrix with this culture media, it's really designed to promote growth of the epithelial cells. So the immune cells die off and the stroma usually dies off within a day or 2. And within 3 to 14 days, can't predict it, organoids will form. And when my first graduate student made this cute little biorender schematic, there was an exclamation point after organoids, because I don't know, everyone, when they have their first organoid model form, they're like, oh my gosh, look at this, it's so cute. And so even though my husband says, I don't know, this is kind of boring, Christy, but he's a scientist too. It's okay. He studies cardiomyocytes. They beat. I get it. It's more interesting. Okay, fine. But so this is what organoids look like under 4x magnification under a microscope. They can either be these cyst-like structures that are on the left here, kind of like a beach ball with no cells filled in. I would say this one here is probably about 200 microns. So our cutoff for an organoid versus cells that are clumped together is about 50 micrometers. You can also have organoids like this one here, some of these that the cells fill the lumen of it. You can have a discohesive structure. And most disconcertingly, I cannot predict based on the stage, the grade, the histology, what the organoids are going to look like under a microscope. So it's something I'm working with a computer scientist on to try to do better predictions. I suspect it's something molecular, but we're working toward that. Okay, so a little bragging, I suppose. So we've created about 150 organoid models to date, but like I said, these do not all grow in perpetuity. Most of these, we use them, we exhaust them, and then we create new models. Our success rate's about 70-ish%, and this really depends on how long the tumor sat out before it went to pathology, how many viable cells there are in the model once we go through the digestion process. And so more of a logistic technical part than based on the aggressiveness of the tumor. And I will say we've had a little bit better success with endometrial versus ovarian, but we've created a lot more endometrial models than ovarian models just by sheer bioavailability of the tissue. So we can do primary tumor, we can do metastatic tumor, we can do ascites cells. For a few cases, we've been lucky enough to get recurrent tissue, which, as our GYN oncologists know, surgery isn't typically common, but we do have a few of those cases. They range stage grade histology. We're trying to get samples from all patients so that we're representing all races, ethnicities. Rurality is a big research interest at University of Iowa. So if there are specific patient populations that I can tap into through collaborations, I'm always happy to collaborate to try to get tumor tissue. We're also creating models from normal, adjacent, uninvolved areas of the endometrium or the contralateral ovary so that we can test safety of drugs. And then the great thing is we have patient outcome data access to that. So we can say, okay, what has the patient been treated with? All right, let's treat the organoid model with it and show that we're replicating what's happening in the patient. All right, now let's try some better treatments. Let's try some novel ideas to increase sensitivity. So what I'm going to talk about today is early stage grade endometrial cancer. So stage 1A, 1B, and then grade 1, maybe grade 2. And so this is typically confined to the lining of the uterus here, just right where a baby would implant. And this represents 80% of all endometrial cancer diagnoses in the US. And the treatment for it is surgery followed by observation, and that's curative for most patients. But there are an increasing number of patients for which surgery is just not a great option. They have comorbidities that preclude safety in surgery. And there are more patients that are being diagnosed at a younger age that desire to preserve fertility. And if you're taking out the uterus, then you have limited that patient's options for fertility. I will point out that the precursor lesion is, it used to be called atypical endometrial hyperplasia. It's now referred to as endometrial interepithelial neoplasia. Please don't change it again on me. This is 8% of cases, 8% of these cases, if untreated, will progress to endometrial cancer. And oftentimes, when a patient has hysterectomy to remove the tumor, they're in 30 to 50% of those cases, or sorry, not the tumor, the precursor lesion, there's concurrent endometrial cancer. So it's hard to, a pathologist will tell you it's hard to distinguish between the two on biopsy. And so really what's important about this type of cancer is it's driven by hormones. Okay. So for the gentleman in the room, let's go through the female menstrual cycle. So in the menstrual cycle, of course, the tissue is undergoing this really dynamic process of degradation, regeneration, and differentiation. And this is directed by two ovarian hormones. In blue here, estrogen, and then in what color is that? Orange, progesterone. So during the proliferative phase here, the spike in estrogen signals for proliferation of the endometrium, obviously, and not only of the endometrium, but also of the stromal cells. And then during ovulation, which is when the egg is released from the ovary, that induces a rise of progesterone here, seen by this uptick. And then at the same time, estrogen levels start to go down. And during the secretory phase, when you have your higher levels of progesterone, you're having modifications of the endometrium that includes decidualization of the stromal cells and differentiation of the epithelial cells. Okay. And let me just check my notes. Okay, that's all I wanted to say about that. But really what I want to highlight is that there's this balance between progesterone and estrogen. And women that are premenopausal and have normal cycles do not get endometrial cancer. Okay, gynocs, correct me if I'm wrong, but I feel like that's kind of a bold statement, but it really is true because that lining gets shed every month and any potentially cancerous tissue is shed. But there are a lot of cases where this balance is disrupted and you have hyperestrogen signaling that is not countered by that differentiating effect of progesterone. And I will say for anyone here that knows breast cancer, progesterone in the breast is different than progesterone in the endometrium. In the breast, it's thought to drive a proliferative signal, whereas in the endometrium, it's driving a signal for differentiation. Okay. So we asked a few years ago, what targetable pathways are modulated by progesterone in endometrial cancer, and can we co-opt those pathways for a potential treatment? So I talked all about organoid models and how great they are. Well, so here's one of the two cell lines for endometrial cancer. It's a hormone responsive cell line. And we just simply treated the cells with progesterone, a saturating level, and did a basic RNA-seq analysis on this. So the three different lines are just three different replicates. And you can see from this, this clustered heat map that there are definite changes that occur in response to progesterone in the endometrial cancer cell lines. And so we did ingenuity pathway analysis. We, as in a colleague, did ingenuity pathway analysis. And we, what I'm showing you here are some of the pathways that were downregulated in response to progesterone treatment. So some of them are expected, like cell cycle pathways like G2M checkpoint regulation, and ER signaling is downregulated. We know progesterone working through its receptor, progesterone receptor, downregulates ER as well. But some interesting ones that we saw come up were PDGF signaling and then JAK-STAT. And so these pathways are linked. So in addition to PDGF receptor signaling through PI3 kinase, it can also signal through JAK-STAT and coordinate with transcription factors in the nucleus to promote cell site or genetic or gene expression changes. And so all of those stars here indicate genes that we saw were downregulated in response to progesterone. So we went into our bank of organoid models and said, can we recapitulate some of the data from the cell lines? So these are 4 different organoid models that I'm showing you here that all have in the primary patient tumor high expression of estrogen receptor by immunohistochemistry. So that's the brown staining within the glandular epithelia. They're here. And whereas across the models, they had a differential expression of progesterone receptor, the target for progesterone. So this one has really robust expression, whereas down in this model here, it's really, there's nothing there beyond the levels of the stroma. And then through just simple qPCR in the models that had high PR expression, we saw downregulation. We validated the downregulation of this PDGFR stat, JAK-STAT pathway, whereas in the models with lower PR expression, we didn't see that similar downregulation. So we asked, if we use inhibitors of PDGF receptor, JAK, and STAT, can we abrogate growth of the organoid models of endometrial cancer? And so going back to the same pathway, we use cediranib as a multi-tyrosine kinase anti-angiogenic agent, mostly because it was being studied at the time in the NRG GY012Q. And we had another paper on cedarenib being superior to bevacizumab, but unfortunately it's kind of gone out of vogue. And then we used a JAK1/2 inhibitor. I'll let my clinical colleagues pronounce these drugs. And then tofacitinib is a specific JAK2 inhibitor. And then we also used a STAT3 inhibitor. And so in this model, which has high progesterone receptor expression, we saw cedarenib alone decrease growth in the organoid model by 50%. So on the y-axis here is cell viability. So a decrease in the bar means we're inhibiting growth. We didn't see a really pronounced effect, even though, stats say yes, but for the JAK-STAT and inhibitors. But when we combined them with cedarenib, we saw that really the cedarenib was driving it, indicating that this is all part of the same pathway. And so then we went on and did this in multiple different models. And what I really want to draw your attention to, well, I want to make your eyes cross, I'm kidding, is that in the models with the lower PR expression, especially this one, we were able to decrease growth of the organoids. This one was fairly resistant, but this one also we saw decreased growth with the combination of the PDGF receptor and the JAK-STAT signaling pathway inhibitors. So what this suggests is that this pathway inhibition can be considered for early stage grade endometrial cancer. And I would love to talk with clinical colleagues about advancing this further. So shifting gears a little bit, we ask, are there other ways to mimic progesterone's actions in the uterus? So to go back to the idea of this imbalance in estrogen and progesterone, we said, are there other agents that can mimic progesterone action? And there are progestins. Okay. These are synthetic progesterone or testosterone derivatives. And we are not the first to have this idea. In fact, progesterone, sorry, the font is a little off. Well, forgive me. Progesterone was actually first discovered in 1933 at the University of Rochester. I put some notes here because I think the history is really cool. And it was called progesterone because it's a progestational steroid. It was successfully isolated and it was shown to maintain pregnancy. Okay, and it wasn't long after that it was synthesized from yams. But that was kind of an interesting little factoid. And then Kelly and Baker were the first physicians to treat a patient with progesterone way back in 1950. And 61 was the first report of tumor regression with progesterone treatment. But progesterone, if you give it as a treatment, would have to be given every 12 hours because it's really unstable. So in the 1970s-ish, Bert and Malley discovered progesterone receptor. And his group found that they first called it a progesterone binding protein. And then they reported that it localized the nucleus where it acts as a transcription factor. And this really opened the field of steroid hormone receptors as transcription factors. And then in the 1970s and 80s were really a time of protein purification, molecular cloning, understanding more about progesterone receptor activity, basic biology of it. And then in the 90s, we started moving into more clinical trials because by that point we had synthesized synthetic progestins that have a much longer circulating half-life than native progesterone. And I will say these studies here in the 70s and 80s and 90s, you have one of the leaders, Dr. Carol Lang, who did a lot of this work. And I will let her speak about her work. I won't try to rehash that. But PR was identified as a predictive biomarker for progestin therapy through the Gynecologic Oncology Group trial, GOG 81. And then my mentor, Kim Leslie, was part of GOG 119. These were the first attempts to maintain progesterone receptor expression by combining it with tamoxifen. And then in 2014 was the publication of GOG 248, which was the first time to combine hormone therapy with a molecular therapy. This was not based on signaling pathways. It was what targeted agent showed efficacy as a single agent. Let's add it to hormone therapy. And I think this is really a cautionary tale because patients had embolism venous thrombosis. Is that right in it? Is that right? Yeah. And so it really highlights that as basic translational scientists, we need to understand how the drugs interact. We were just talking about this at breakfast this morning before we start treating a patient with it. And then the more recent trial, NRG GY011, established Ki67, so the proliferative marker as a biomarker for response to progestin therapy. Okay, so I hope I've convinced you that progestin therapy is great, and 70 to 80% of patients initially respond to it based on the specific progestin they're given, because there are 3 different ones that are used clinically. And in a study this is a study from Finland, an observational study of women who were treated with progestins for contraception. They saw a decreased incidence of endometrial cancer, lifetime incidence, more than 50% decrease. You're not used to looking at odds ratios. This is what it is. So this means it's a 54% decrease in incidence of endometrial cancer in women that had progestin contraception IUD, decrease in ovarian cancer, lung cancer, pancreatic cancer. What in the world? This is a little unusual. But yet, despite that, hormone therapy really is a small part of the market in using cancer therapy. So on the left here, I'm showing you the market share for different types of anti-cancer agents in 2020. So largely angiogenesis inhibitors like bevacizumab, but PD-1 inhibitors are starting to come on board. This little teal bar here is endometrial cancer. And by 2029, you can see that immunotherapy is really projected to dominate the field, and that teal bar is getting even smaller. But we think that there are opportunities to reverse this market share with great data showing efficacy. So there are some reasons why progestins aren't used more often. So I told you that patients respond really well initially, but recurrence is really common, anywhere from 20 to 40% after the initial response. So then we said, can we reverse resistance? And one of the major drivers of endometrial cancer is estrogen, that excess estrogen stored in adipose tissue. And so in women that are obese, both pre and postmenopausal women, or those that have dysregulated estrogen, like in polycystic ovarian syndrome or in ovulation, we have an excess of estrogen that's not combated by progesterone. But anyone tell me the new drug on the market right now for obesity? GLP-1 agonists. So they came on the stage and we said, what happens if we combine GLP-1 agonists with progestins? So like any good scientist, we went to the literature, and people had shown in cell lines that GLP-1 agonist will increase PR expression. And then here are 2 different studies from other groups where GLP-1 agonists as a single agent, were used to treat endometrial tumors in mice. This one is a model of a low-grade endometrial cancer in an immunocompromised mouse. You can see exenatide really is effective at maintaining the tumor growth. And then this is a more recent study from Vicky Bay-Jump's lab where they put mice on a high-fat diet. This is a genetic model of endometrial cancer. I will say it's a high-grade model with a P53 LKB1 mutation, so not quite mimicking the early stage grade endometrial cancer, but still great work showing that tumors are larger in the mice with a high-fat diet, and they can be controlled with a GLP-1 agonist. So of course we went to our organoid models and said, what happens if we combine a GLP-1 agonist with a progestin? And in this case, we're using levonorgestrel. This is the Mirena IUD that millions of women use for contraception around the world. And we see not a significant effect in this model. So again, like the data I showed you earlier, on the y-axis is cell viability. So you want to see a decrease in it. But across different models, we see when we combine the 2, an overall decrease in viability compared to control. And I will note that in most of these models, levonorgestrel at the dose we use doesn't really seem to be affecting growth at all. And this work is being pursued in a SPORE project led by Kim Leslie and Andrea Hageman at WashU. Novo Nordisk is providing GLP-1 to combine with a Mirena IUD for conservative management of endometrial cancer with a secondary goal of fertility. right, so there's another reason though why progestins aren't used more often, and it was this pesky, wonderful study called the Women's Health Initiative. And in 2001, the results of this study came out, and there was a 1.2-fold increased risk of breast cancer. This is for women that are on progestins for postmenopausal hormone therapy, not endometrial cancer. And so this caused a considerable fear in the public, because you hear 1.2-fold increased risk. No, please don't give my mom something that's going to increase her risk of cancer by 25%. And so, as you can imagine, the media frenzy was crazy. And you talk to you hear talks from the investigators of the Women's Health Initiative, and they say it got completely blown out of proportion. And why is that? Yeah, it's a 24% increase in relative risk, but a 0.08% increase in absolute risk. So what does that mean? For every 10,000 women, 8 more have an increased risk of breast cancer per year. So really, you're going from 33 cases per year per 10,000 to 41 cases. And through a lot of, there are also other issues with the Women's Health Initiative study design, which I won't go into today because I want to get to the rest of the data. I'd be happy to talk about it. But just 3 weeks ago, the FDA removed the black box warning from hormone treatment for menopause. They remove references of risk to cardiovascular disease, breast cancer, and probable dementia. And they're recommending to start HRT within 10 years of menopause onset, but before 60 years of age. And that the benefits outweigh the risks at this point. So we were ecstatic, because that means that the door is open for progestin therapy being revitalized for endometrial cancer. And so our hypothesis is that the ideal progestin therapy will both inhibit endometrial cancer growth, but also minimize adverse effects on the breast and the normal uterus. And so this is a project that is in collaboration with investigators at University of New Mexico, Utah, Kansas, and Iowa. And so what I'm going to focus on, the project that I lead, is how can we repurpose progestins to inhibit endometrial cancer growth. So we are using progestins that are FDA approved, passed the bar for safety for other indications, contraception, hormone replacement therapy. I think it's really important to note that we are using the same doses that patients are receiving for contraception. So I put together a cute little word cloud. Millions of women around the world are on contraception therapies, and they are testosterone derivatives, progesterone derivatives, spirulactones. They're all different structures. And so, but yet there are only 3 of these drugs that are used for endometrial cancer treatment. And we said, what if we go to this panel of progestins that are used for other indications, and can we repurpose those as treatments for endometrial cancer? Some of the newer ones, like drospirenone, which is a spirulactone, is used for contraception because it has less androgenic effects like unwanted hair. Maybe that lack of AR activation would have benefits for endometrial cancer. So what my lab does is we have a live cell imaging system here that we can do high throughput screening of drugs. So this little box here is an incubator. I call it the cell hotel. You can put in 8 different microplates at a time. So nothing makes me happier than to walk in on a Friday afternoon and see that my students are collecting data all weekend long. And it runs on a scheduling software that fetches the plate from the biospa, that's the cell hotel, and then puts it into the plate reader, where we can analyze different features. We can look at growth. We add in fluorescent dyes to monitor different phenotypes in response to drug. So essentially what happens is you treat with drug and you add in fluorescent dyes that can, for example, annexin 5 to look for apoptosis, or indicators of cell health like cytotox screen. And then you program the imaging software to take serial images along the Z plane. And this is done through the biospa citation system. I promise this is not I've asked them to give me a cut on products, but anyway, they won't. Anyway. And then we have these serial images along the Z plane, which we then smash together and make a Z projection that we can then analyze. And so what a graduate student in my lab, Katie Culling, has done is gone to the list of progestins that are used for contraception and tried those, pick some from each of the different structural categories. Don't ask me about the structural categories. I partner with structural biologists and chemical experts in trying representatives of these. So it's not a live cell imaging talk without a video. So in red here, you'll see apoptosis, an exon 5 staining. And this is at time 0. And I will say, just through virtue of the manipulation process to plate the organoids for live cell imaging, you have some cell death. So, and we're able to calculate that, the red here. Okay. But this is just a normal vehicle treated organoid model that's growing over time. You can, pick one and focus on, you see it's getting bigger. The video is going to loop. Sometimes the organoids migrate. This is a pretty static model, but you can watch them grow over time. Kind of fun. Then we added drugs. So we add progesterone, since that's our baseline. We want to mimic progesterone. And here you can see that most of these organoids are not growing and they're getting a red center, which means, interestingly, they're undergoing apoptosis. Although there's some that you can focus on, like here, where they're not all responding. And I think this is really fantastic because it's capturing the intratumoral heterogeneity in response. So then we added levonorgestrel to it. This is one of the hormones that's used for endometrial cancer treatment through the IUD or through oral. And you can see, just like progesterone, lots of red appearing and the organoids are not growing. So this is great, but I can't always publish a video, and for an NIH grant, you can't submit videos. So we have to have a way to quantitate this. So this is the original Z-transformed image, and then we apply a primary mask, which is based on object size. So I said we classify organoids as anything larger than 50 microns. So we've eliminated that, and this will also get rid of some of the debris that might be there. And then we apply a secondary mask, which is in pink, for circularity, because sometimes you might get, I don't know, like blobbed out cell structures, and you don't want to include those. And so then we can calculate pixel intensity within the mask and then fluorescence intensity within the mask. Everybody follow this? Okay, good. And so then we get a growth curve like this. So these are a panel of drugs at one concentration, and then we do dose responses of them. So we always use starsporin. This pink line here is our positive control for cell death. And then these are the different progestins. And all I really want you to appreciate is that they're different. They're not all performing the same. This was something that we were relieved to see, that this is not a class effect, that all progestins act alike, that they have different properties. And then some of them induce apoptosis. And I will say, in general, the trend is that if they inhibit growth, then they induce apoptosis. But there are some that are inhibiting growth, but they're not inducing apoptosis, which is also interesting. All right, so how do we take these data and we combine 11 different drugs across different doses, time, in models? So the way we do that is this is just a little schematic. We take the slope of each line. If it's not linear, then we log 2 transform it. And this is just by using simple linear regression, we determine the slope, which is the growth rate. And then we can use the growth rate normalized to vehicle to determine the Z-score, which captures the effect size. So for those of you that like math, this is the actual formula. This was created by our colleagues, Jay Gertz at Utah and Miles Pufall, part of the hormone therapy PO1 group. So basically the normalized growth rate is the growth rate for the drug at each dose divided by the vehicle. And then the Z-score is the average of the normalized growth rate subtracting DMSO and then divided by the standard deviation. And so this is how you get the effect size. And so a really hardworking graduate student in my lab, Katie Culling, has taken she's done this in 14 different models now. I think this is the most recent data, where she's compared all 11 drugs across the models at different doses. So any bar with an X is just indicating there wasn't enough sample to test that drug in that model, which is a clear limitation of organoids. They are ordered based on their structural categories. So we have native progesterone here, hydroprogesterone here, medroxyprogesterone acetate, and that's MPA, and megase or megestrol acetate. These are two commonly used agents for endometrial cancer treatment. Retroprogesterone, and so on and so forth. And what's really interesting is just by eye, you see that the testosterone derivatives, levonorgestrel, northistrone or northindrone, and then northistrone enanthate, which is a prodrug of northistrone, they all seem to have the most robust effects across all the models. So I'm sorry, I should have said you're looking for red. A decrease, red is the desired effect, a decrease in growth. Blue is that the drugs are promoting growth, which obviously you don't want to do. We've also done this for apoptosis. And so these data are feeding into an algorithm to rank the progestins and their efficacy, but that's not quite ready yet. So. But what Katie has also done is look at what are the gene expression changes that occur in response to the different progestins. So again, getting to that idea, do all progestins perform the same or are they different? And so this is the heat map here showing, this is for one model, 14133. It's apparently her favorite model because it grows really well. Has these beautiful cyst-like organoids that are very easy to mask in imaging. And you see a differential response into by growth. And when she performed RNA-seq, she first looked at progesterone receptor gene expression. So we know that progesterone decreases progesterone receptor. This is a normal ligand-mediated downregulation so that we don't have sustained levels of PR. And all of the drugs seem to have this canonical PR downregulation, which is reassuring, because they're working on target. But interestingly, FKBP5 is a canonical PR transcriptional target. We see heterogeneity in their response. So for example, MPA is really increasing expression of FKB5, but drugs like Dienogest aren't. So we're in the process now of expanding these data out to many, many more models, spending lots of money, and trying to understand, can we use this to more intelligently identify which agents to advance into clinical trials in patients. And so the last thing I'm going to talk about is just a very brief couple of slides on our efforts to repurpose progestins in the recurrent setting. So patients that have already presented. So this is a case study of a patient that had a very high BMI and a history of EIN, the precursor lesion. And she had been prior treated with megase as well as northindrone, because the most common symptom that patients come in with is abnormal bleeding, postmenopausal bleeding, or off-cycle bleeding. She underwent surgery to remove the uterus, and that at surgery, the diagnosis was stage 1, grade 1 endometrial cancer, so that low-grade endometrial cancer. So a graduate student in my group, Abby Morrison, diligently created organoid models from this patient's tumor. And so on the left with the vehicle, you can see it's kind of a slower growing model, and it's quite migratory. So the video kind of moves around, jumps a little bit sometimes, just a rendering effect. But like this model here, you can see it kind of wiggling around. This, I will say, this makes it a little bit challenging to for the data analysis, but we're still able to do it. And then when she treated with very high doses of the drugs that the patients were treated with, she saw that the model still continues to grow. So this is confirming the resistance in the model. And so then Abby took some of the drugs that Katie had found to be most effective in the primary setting for endometrial cancer and treated that recurrent or the resistant model with it. And so this is just a video of one of the drugs, NETE, northistrone, enanthate. And you can see that it is definitely decreasing the growth. She's got in an exon 5 dye for apoptosis, as well as a cytotox green dye for cytotoxicity. I will say this dye is not fantastic. Where if anyone has ideas for better dyes, let me know. And then she then quantitated this here, and really it was NATE that seemed to reduce growth almost to levels of staurosporin. And so she's expanding out more models. I will say it's been a little bit more challenging than we anticipated, getting models of patients that have progressed on progestin therapy. But with some new clinical trials and observational studies that we're opening up at University of Iowa and our partnering institutes, we're hoping this project will advance a lot more quickly. Okay, so, I'm great on time. All right, so to conclude, hopefully I've convinced you that inhibition of the PDGF receptor JAK-STAT signaling pathway is an alternative approach that we can phenocopy the effects of progesterone in endometrial cancer. Or we can combine a GLP-1 agonist with progestins. And I mentioned that this work is already moving into clinical trials in patients. We're also trying to repurpose progestins for other indications as a non-surgical approach. I think this is really important that we are exploring options for patients that surgery isn't a great option. So just because, hysterectomy has traditionally been curative doesn't mean that that's what can always be done in the future. And we found that some of the newer progestins are more potent against organoid models of endometrial cancer. And we benchmarked everything against MPA, which has been used in clinical trials the most. I will say there are some class action lawsuits against the makers of medroxyprogesterone acetate MPA because as a contraception, so Depo-Provera, the shot, it's increasing the risk of meningioma. So we are not moving forward with MPA in our subsequent trials because we want to not jeopardize enrollment. So what are we doing next? Well, we've created a pipeline for clinical testing of progestins where we can pre-screen them in the organoid models and then move them into a surgical window trial, which Dr. Erickson has run some of those as well. And the idea is those that seem to be most effective at creating a biological effect in the surgical window trial, which let me explain that. At the time of biopsy for diagnosis, you put a patient on, they're usually scheduled for surgery in about 3 or 4 weeks. And so you put a patient on a drug during that time. So you can get a pre-treatment and post-treatment patient match sample and analyze different biomarkers of response. This is not an efficacy therapeutic trial though. But it can really be used to better guide which agents to advance into a therapeutic trial. And then also, I think there are some really great opportunities using these models and our collaborations to assess the efficacy of newer progestins in both the primary and the recurrent setting, as well as in combination therapies like the GLP-1 agonist. And in fact, when we are setting up the upcoming surgical window trials to try out some of the progestins, we debated, do we have GLP-1 agonists as an exclusion criteria? And I think that debate is still ongoing because a lot of patients are coming in on GLP-1s. Yeah. Okay, so with that, I really just first and foremost, I always acknowledge the patients that donate their patient tumors for research because without them, we couldn't have done anything that I've shown you today. Well, maybe the cell line RNA-seq data, but really that's what's driving our research program. I have the privilege of mentoring a really fantastic group of students at the University of Iowa. Most of the work I showed you today was led by Katie Coling here. She's a 4th year graduate student and hopefully she'll never graduate. And then Abby Morrison is working on the resistance project and is really making some cool progress on it. I do not specifically recruit women to my lab. I promise there is no discrimination going on. I think it's just the disease and that we study. I have benefited from mentorship from Kim Leslie, and now it's kind of exciting to call her my collaborator and partner, as well as Miles Pufall at the University of Iowa, who's also been a secondary mentor to me. Jay Gertz at Utah, Christy Hagan at Kansas. She's leading the breast safety studies. There's some really sophisticated models. Pilar de la Puente at Stanford, Eric Prosnitz, Mara Steinkamp at also at University of New Mexico, and Tian Li Wang at Johns Hopkins. So work I didn't talk about is we're moving these agents also into genetic models of endometrial cancer. So Tian Li's group has developed really the best to date that replicates early stage grade endometrial cancer. And then Mara is has developed humanized mouse models of endometrial cancer. This work would not be possible without funding, and I hope that continues. And then my lab QR code is here. Feel free to email me. Okay. All right. Questions?
I have microphones for anyone who has a question.
That was great. Thank you very much. So as an AR biologist that studies prostate cancer, I take great offense to you calling FKBP5 a PR target gene, because we studied as an AR target gene. Those that study glucocorticoid receptor signaling study as a GR signaling molecule. So when you create these organoids, my question is, what repertoire of steroid receptors do they maintain, and does that maintenance of the steroid receptor repertoire match what was originally in the tissue? And then how do you interpret the steroid receptor through which a lot of these steroidal compounds are acting? You're interpreting it as though it's acting through PR, but a lot of these effects could be explained by actions on other steroid receptors, or the kind of the whole gamut of steroid receptors competing for each other. So that's the first question. And the second question is, how do you determine the doses of these drugs that you're treating with? Because it looks like you're doing all of your work with single fixed doses, and you can have great differences in effects there.
Did Miles Pufall from Iowa put you here to ask these questions? So actually, as part of the PO1 here, I have a little schematic of it if anyone's interested. Miles is studying the mechanistic aspects of the different progestins. So absolutely, these progestins are not exclusive to PR. And in fact, we hypothesize that some of their beneficial effects or detrimental effects may be through their off-target, I'm careful using off-target these days, but it may be through their actions with other steroid hormone receptors, whether it's good or bad, and maybe even in the breast too. Maybe that's what's promoting this increased breast cancer risk. We are in the process of characterizing steroid hormone levels in our organoid models through the RNA-seq data. I know protein is better. We really have to prioritize which assays we do first, because sometimes the models just become exhausted. And so we will go back to the patient tumor tissue for a surrogate, and so we try to prioritize assays that we can get the most data out of it. So we're doing RNA-seq on all of the models. We're also doing whole exome sequencing on all of them to bend them into the molecular categories for endometrial cancer, which I didn't talk about today. I didn't want to muddy the waters too much. So yeah, that is coming. And by the time the paper gets submitted, those data will be there. But I think it's really an important point.
And to what extent do you have access to the tissues from which those organoids were derived to do those head-to-head comparisons? Are you doing that for all, a subset?
Yeah, so we have access to archival FFPE tissues for all of the models. Oh, and your question about drug dosing, this is something we talk about all the time. Do we go based on the Cmax in circulation? What is the actual drug level in the endometrial tumors? So we do a dose response for all of them, from 1 nanomolar to 10 micromolar dose response. And so that heat map, that red heat map that I showed, actually takes into account the full spectrum of the dose response curve. Yeah.
Any other questions in the room?
So for all the researchers in the room who need tissue, I think your universal IRB is like the best idea ever, and that's what we should be doing for sure.
That's not my idea.
But the fact that you got it going and had this, I mean, that's the number one thing that we need, I think, in this institution is a universal IRB. We run into these problems all the time. We can't consent patients prior to surgery because there are 50 million people in the room. So after patients consent, so you get them to consent for the tissue, do you have to write an IRB then again to use that tissue? So it's one universal IRB and you can use those tissues however you want.
Yes. Let me explain the history of our universal IRB. Our former cancer center director, George Weiner, he launched the Comprehensive Cancer Center in Iowa. He was very interested in the Orion initiative. It's the collaboration between Orion, Moffitt, and M2Gen. Orion, I always forget what it stands for, but it's like a research exchange network where you provide tumor tissue to Orion. They do RNA-seq and whole genome sequencing on it. You also give them full clinical data and update. And each university that contributes has access to all of the data that come out of the sequencing. And then through a collaboration, you can also partner with other institutions that are contributing to Orion. That's why I ask where you're getting your data, your sequencing data from. You can partner with other collaborating institutions and get sequencing data, for whatever project you want to study. But in order for us to become part of Orion, we had to have a universal IRB that was pan-cancer and allows the universal consent. We do still have a gyn-onc biobank. We have a really great women's tissue repository that goes beyond cancer through placenta, throughout the lifespan of the woman and partner and baby. And so we do have a separate IRB, but we're in the IRB is in the process of just adding our gyn-onc IRB as an amendment to the universal IRB. Does that make sense? Yeah. No, it's been, this was definitely a Herculean effort by Dr. Weiner.
Fantastic.
I'd be happy to share the documents if anyone wants
We'd
them.
love that. Thank you. Thank you. Any other questions from anyone?
Okay. Thank you for doing this work. It's so important. Actually, endometrial cancer and steroid receptors and hormones are so understudied, considering this is like a hormonally active cancer. So I'm so glad you're doing this. It's really great. As you look at all these new progestins, it would be great to replace MPA, these things. Do you think, though, that they're really going to overcome primary progestin resistance? Or is it just that it'll be a better drug with better side effects? Like, are there some, do you have a signal that tumors that are non-responsive to progestin are maybe we're going to make them responsive with some of these newer agents? Or is it just a matter of kind of range of efficacy and tolerability of the drug?
That's a great question. And I really think we're just going to have to see what the data show us, honestly. I mean, the only reason I think we might be able to overcome primary resistance with alternative agents is maybe they're acting through different steroid hormone receptors, different activities. But, I really think about the safety, right? Like if the goal is to preserve fertility, for example, what kind of treatment can we give that would still allow that?
All right, we've got one final question from our online audience. Doug Yee asks, the rising incidence of endometrial cancer has been linked to insulin resistance. Have you looked at insulin signaling or hypoglycemia in the organoids?
The short answer is no, but definitely that's an area of research that we'd like to expand into.
All right, I think we're out of time, so let's say thank you one more time to Dr. Thiel.